Hello, this is Digestive System video number 7. In the last video I told you the next video will be the large intestine, but I lied. I decided to instead focus on two of the accessory organs, the gallbladder and the pancreas, and two of the main secretions that are being secreted into the small intestine, specifically the duodenum. Those include bile, and that's from the gallbladder, also from the liver as well. And the pancreas is making pancreatic juice. So those are important for digestion. We'll talk about both the organs, gallbladder and pancreas, and the secretions from them. The following video will cover the liver, and then we'll end with one video talking about the large intestine. So let's talk about bile first. Bile is a secretion product that is originally made by cells in the liver. The hepatocytes in the liver make bile. It is stored in the gallbladder. And then when needed, if you eat something, especially something containing lipids, then the gallbladder will release the stored bile into the duodenum. The gallbladder is, so it's kind of a temporary storage location. Bile is a kind of a yellowish green fluid, and it's also alkaline, and it contains a bunch of different components. So they're listed on the slide here. Bile salts, and bile salts are amphipathic molecules, meaning they're partly hydrophobic, partly hydrophilic. There are bile pigments, and those are from the breakdown of heme group and from blood. There's cholesterol, there's neutral fats, phospholipids, and mix in a bunch of electrolytes as well, various ions, excuse me. So bile is mostly lipid, high lipid content with a few electrolytes mixed in, and bile salts, which are amphipathic. Now, the bile salts are important for the digestive function of bile, and that is aiding with the digestion of lipids. And the main thing that the bile salts do, since they're amphipathic, is that they emulsify lipids that are present in the chyme. So if you chew up a meal and it gets through your stomach, it's going to be homogenized, except for the fact that the environment in the digestive tract is aqueous, and any lipids are going to form into these large globs in an aqueous environment, right? They'll separate from the aqueous environment. So emulsification is when the bile salts interact with these large lipid globs and disperse them into smaller globs. All right, so it's just separating large aggregates of lipids into smaller, more accessible for digestion globs. Okay, so that's one thing, emulsification, and by virtue of emulsifying lipids, this is going to help speed up digestion and also help with lipid absorption. So let's look at exactly how emulsification works, and this slide is illustrating a large fat globule that has reached the duodenum. This would be the status as it goes through the stomach. And so this is going to be really difficult and slow to digest because digestive enzymes will be restricted only to the outer surface of this globule. So what we want to do is to break this down to smaller spherical globules, and that's where the bile salts come in. Bile salts are being amphipathic, have one side which is hydrophobic and one side which is hydrophilic. So the bile salts interact with their hydrophobic side toward the globule and their hydrophilic side toward the aqueous environment inside the lumen of the digestive tract. And by doing so, they allow these smaller globules to break off and travel through the digestive tract. So now these are much more accessible. There's more surface area from a bunch of small globules from a single really large globule, and that gives more access to digestive enzymes. And that's basically what emulsification is. How is bile transported? So I mentioned that it's made by the hepatocytes in the liver and stored in the gallbladder and then secreted into the duodenum. So that's kind of a complex pathway that's involved. Basically, there are a number of ducts connecting the liver to the gallbladder and the gallbladder to the duodenum. So these have different names. Common hepatic duct, for example. Hepatic is referring to the liver. And so there are basically small pathways, one leading from the liver and one leading from the gallbladder, and these merge into what's called the common bile duct. So let's look at the pathway shown here. Here are the ducts coming from the liver. That's where bile is made. So there are a number of ducts coming from different parts of the liver, and they're going to merge into what's called common hepatic duct. Here's a duct coming from the gallbladder. Gallbladder is storing and concentrating bile. And this is called the cystic duct. Cystic duct merges with the common hepatic duct to form the common bile duct. And then this is going to carry bile and connect into an opening into the duodenum. And this opening, by the way, is also going to carry secretions from the pancreas. So this opening is called the the duodenal papilla. And so you can see that here. And that's the opening for both bile entering into the duodenum and the pancreatic secretion called pancreatic juice as well. All right, so that's the pathway that's involved. A little bit about the gallbladder. So the main function of the gallbladder is storing and concentrating bile and secreting bile when chyme reaches the duodenum, especially lipid-containing or lipid-rich chyme. It's a relatively small structure, and it's a muscular sac, and contraction of the smooth muscle in the sac is what basically squirts out a glop of bile. It's associated with the liver very closely, and it kind of protrudes from the ventral surface of the liver. And it releases bile into the duodenum. Now, one problem that can go wrong with the bile is the formation of what are called gallstones or cholelithiasis. And this is a gallbladder that has been removed, showing collection of a huge number of gallstones. So since one of the main functions of the gallbladder is to concentrate bile, this is a problem that can crop up. Gallstones are usually crystal formation, solidified bile salts, cholesterol, calcium, and maybe other electrolytes. They can be small or really large, and you can see here numerous pretty large gallstones. The main problem with the formation of gallstones is blockage of the cystic duct, which causes an unusually large accumulation of bile in the gallbladder and creates pressure and pain. These can be broken up by ultrasound, but in something like this, a pretty extreme case, the typical thing to do would be to remove the gallbladder, and this is called cholecystectomy. Okay, you can live without a gallbladder, and the liver will still make and secrete bile. It's just not as well regulated as if you had a gallbladder. So there's, usually some problems initially after having a cholecystectomy, but you can live just fine without it. Some of the risk factors for developing gallstones are listed on the slide: old age, obesity, Caucasian, and kind of being a couch potato, lack of physical activity. Females more likely than males. And it is asymptomatic unless the duct gets blocked, and that causes inflammation and pain. All right, let's go on to the pancreas. First, a little bit about the pancreas itself, and then we'll talk about the main secretion product of the pancreas, which is pancreatic juice. Pancreas is located deep to the stomach, so especially the greater curvature of the stomach. It is a retroperitoneal location. And the head of the pancreas, and I'll show this on the next slide, is basically cradled in the duodenum, the curvature of the duodenum. And remember, the duodenum is also retroperitoneal. The pancreas is a gland that has both exocrine and endocrine function. The endocrine function are through the pancreatic islets or islets of Langerhans that make insulin and glucagon, hormones involved in regulating blood glucose concentration. And that's a very small minority of the total number of cells in the pancreas. 1% or less of the cells in the pancreas have the endocrine function. 99% of the cells are involved in the exocrine function. And that is making and secreting digestive enzymes in a mixture of watery alkaline solution, and then that gets secreted through an opening into the duodenum. The cells that are producing these enzymes are called the acinar cells, and the enzymes that they produce are 4 main types of enzymes that are capable of breaking down the 4 main classes of macromolecules. Amylases, involved in breaking down carbs. Proteases break down proteins. Lipases break down lipids. And nucleases break down nucleic acids, such as DNA and RNA. This is showing the gross anatomy of the pancreas. Remember, it's in the upper left abdominal quadrant, and this is embedded in the dorsal body wall, retroperitoneal location, along with the duodenum. Okay? The pancreas has sort of a comma shape to it. It has a relatively wide head, and that's cradled in the curvature of the duodenum. And then it spreads out laterally to this kind of tapering tail, head and tail. If you look interior to the pancreas, you see a set of ducts. There's one main pancreatic duct that's running down the center of the pancreas, and this is going to connect into what's called the ampulla, and that feeds into the papilla, the opening into the duodenum. And then branching off this main duct are all these smaller ducts. All these smaller ducts are going to connect to acinar cells, which are producing the enzymes. So this slide shows the histology of the pancreas. Minority of it is this, the pancreatic islets, or the islets of Langerhans, with the endocrine cells producing the hormones, insulin and glucagon. The majority of the cells in the pancreas are these acinar cells. The acinar cells are making and secreting the enzyme into a duct. And this duct is a small duct leading into the larger and larger duct system. And that's eventually going to be secreted into the duodenum. This is just showing the acinar cells and the main classes of enzymes that it makes, the 4 classes I mentioned. And also that it's making an alkaline secretion, and that's due to the duct cells. The cells lining this duct are making bicarb, and that's going to be part of it as well. All right, the last slide in this video is summarizing the digestion of macromolecules. So we have talked about all of these now, and here are the 4 classes of macromolecules, and here are the different locations along the alimentary canal and where these enzymes are made. So let's go through this a little bit. The oral cavity is producing salivary amylase and lingual lipase. So starches begin to be digested in the oral cavity due to the salivary amylase. The lingual lipase would break down lipids, except this is not active yet. It's secreted in an inactive form and is only activated when it reaches the low pH of the stomach. Okay? There are no enzymes to digest proteins or nucleic acids in the oral cavity. In the stomach, additional enzymes are added. So if you remember, the chief cells secrete pepsinogen, which is inactive. When that hits the acidity in the stomach, that's converted to the active form, which is pepsin. That's a type of protease. Also, this is showing the lingual lipase, which is activated in the stomach. And the stomach chief cells also make gastric lipase, which is inactive until it hits the acidity and is activated. These are two types of lipases. They have slightly different activity and they work on different substrates. Okay, so in the stomach now, the amylase is going to be inactivated when it hits the acidity of the stomach. But you have active proteases and active lipases. Still no active nucleases yet. Okay, the small intestine now is when most of the chemical digestion is going to happen. And this is going to be through pancreatic juice in this row and brush border enzymes in this row. Pancreatic juice has all of the four main types of digestive enzymes. Amylases, a whole set of different types of proteins, pancreatic lipase, and nucleases that are capable of breaking down both DNA and RNA. So whatever you eat, any kind of organic matter, plant or animal, is going to have all of these four classes of macromolecules and can be digested. Now there are some exceptions. Some plant fibers were not equipped to break down, so those will not be digested. Here are the enzymes in the brush border. So remember, the brush border are the microvilli present in the intestinal, mucosal, epithelial cells. So there's a whole host of different types of amylases. They have different names. They're equipped to break down different types of polysaccharides or disaccharides. And there are also a number of other proteases as well. These are a couple that are listed here. There are no brush border lipases, but there are a couple of brush border nucleases. Okay, so those are the main sources for enzymes carrying out chemical digestion in the alimentary canal. So I'm going to end this video here. Next video, we'll discuss the liver.